Hello, my name is Angad Kahlon, and this is my presentation for the Comp 359 final. I'm a part of Group 6 with Jasjeet Singh, and this group was focusing on dynamic programming, which is one of the topics we learned about this semester. Specifically, I'm focused on the DNA similarity scoring project within dynamic programming, and let's get started here. I just want to, before I go into the implementation, I just want to give some background information. Basically, DNA and RNA are what we're dealing with in this project in terms of data. So DNA and RNA are important strands of genetic info for all types of life on Earth. They're made up of nucleotides. Four. There's adenine, which is represented by A, thymine, which is represented by T, and its uracil, which is represented by U in RNA. Cytosine, which is represented by C, and guanine, which is represented by G. And the pairing between these nucleotides are that A pairs up with T slash U, and C pairs up with G. And same thing with vice versa, where G pairs up with C, and so on. Over here, there's a quick diagram of what the structure of DNA is. Um, since I'm focusing on DNA similarity scoring, some important applications of DNA similarity scoring and finding out the similarity between two DNA sequences in the real world are such as understanding mutations, evolutions, and protein development. And in this project, I specifically focus on two COVID-19 DNA sequences, which I'll talk about in a little bit. And for this project, I used a tutorial by Open Source Automation on the automatic.net, and they created a basic algorithm which measured DNA similarity with Python and dynamic programming. And I can quickly show you the website here. And basically, with dynamic programming and their implementation, they come up with this matrix or matrice here. And over here, what this matrix represents, basically in short terms, is that we have one DNA sequence on the top, one DNA sequence on the side, and for each match between the two letters or the nucleotides, we get a they get a plus point. So that little grid over there would get a plus point, and for us, every mismatch they would get a penalty, so it would go down by -1. So like G and T, they don't match, so they would get a -1 score. And a little bit more about the DNA similarity scoring project. What it does is that it takes two different DNA or RNA sequences, it aligns them for maximum similarity of nucleotides, and then returns the similarity and info about the additions, deletions, or mutations. So my implementation specifically deals with a 2023 and a 2024 COVID-19 sequence from the NCBI, which is a biotechnology information online source. But you can take any DNA sequences, even human ones, even animal ones, or plant ones, and you could feed them into my implementation and it should work. And what my implementation does is that it records a score in a matrix. And I actually have shown my matrix right here, my score matrix for the DNA similarity. The example that they gave on the open source automation on the automatic.net, they only have a sequence of around 9 characters, while the 2023 COVID-19 sequence and the 2024 sequence were approximately 100 letters by 100 letters. I just chose like a specific, the last 100 characters, because otherwise it would be too large of a data type. So yeah, I only chose the last 100 characters or so. What it does is that it records a score in the matrix. So this is a matrix full of scores, and for every mismatch, again, a penalty is applied of -1, and every match, you get a reward of +1. And in the aligned DNA sequence that is returned at the end, a dash is in place for every mismatch. So if we talk about the complexity of this algorithm is in big O notation is n times m, where n is the length of the first sequence and m is the length of the second sequence. If the sequences are same length, then we could just do n times n, which is giving us n squared. And n squared also makes sense because we have one nested loop where we have a for loop inside of a for loop, giving n squared again. And we also have a score matrix, which is two-dimensional, which gives us n squared as well. So the complexity in general is n times m or n squared. In terms of the code implementation, my implementation, the code I coded it in Java on VS Code along with JavaScript for the user interface implementation. There's functions for the algorithm calculations and the user interface, and the data structure that I used to represent the matrix was a multi-dimensional integer array. And in the user interface, it asks the user for the sequences, returns the aligned sequences, changes, matches, and the percentage of similarity. And there's also an option to see each iteration if you would like to see the visualization of how the code is take at each iteration of that loop that I'll show in a little bit. So I'll show you the code here. Just a quick overview. So over here, the top is just implementing the user interface. Here I implement the integer multi-dimensional array, and it goes through, and this is the main while loop. And in this while loop, if it goes through three scenarios, if they match, if sequence X has an addition or deletion or a mismatch, and if sequence Y has an addition or different nucleotide, it goes through those three scenarios and then changes based on that. And then the rest is just part of the user interface and returning the data. So if we give this a quick run, we can see here this user interface opens up, and I've already added in the DNA sequence from 2023, which is real COVID-19 sequence, and the COVID-19 sequence from 2024, which is just from April 4th, actually. So here we have a button for visualizing each iteration, or you could just hit run. I'm just going to show you what you can do in total. If you don't click the visualize each iteration, it'll just show you the information. So here showing us that it's on. So if we hit run now, if we start from the top here, this information is reported. This information is also reported if we just hit run. So it'll show you some information. It'll show you the aligned first DNA sequence, and then the aligned second DNA sequence, the number of changes. So there was 12 in this case, the number of matches, which were 91. And so the similarity between the sequences is 88% similar. So basically, the COVID-19 DNA sequence from last year and this year, they're only 88% similar, which shows how fast that virus, the COVID-19 virus, is mutating and changing. And so over here we have, you can also visualize each iteration. So starting with iteration 0, and you can keep on going until the end where you can see iteration 102, where you have the first DNA sequence and the second DNA sequence, and each dash represents an addition or deletion in between the sequences. So if we go back to the presentation here, if we talk about group work, we divided the work by different implementations. Jasjeet did a similar implementation of a minimum edit distance problem, while I did the DNA similarity score. All the code is available on our GitHub, and that's what we used for version control and uploading and sharing the code. We used WhatsApp for communication and helping each other out. And for other work, we used Google Drive and Docs, so we can easily share each other information with each other and documents. In terms of going into the deep, what Jazzy did and how that relates to what I did, this is an example. This image shows my score matrix, and Jazzy also created a score matrix, which is pretty similar, but his is the minimum edit distance matrix. But they're both, at the end of the day, both of them both of them use dynamic programming, which is like breaking down, it says right here, like dividing a large problem into smaller subproblems and working up from those subs, like small subproblems, and the use of that score matrix to align and find minimum edits, or in my case, the DNA similarity. So what Jaji basically worked on is finding the similarity between two strings or words. So this is useful in applications like autocorrect. When you have autocorrect, like if the computer tells you you want to type this word, when you type slightly an incorrect version, and so it finds out the similarity between those two words, which is similar to what I did. It's just Jazzy did the word similarity, and I did the DNA sequence scoring. And so, but the base model of both of them is the same, of using dynamic programming and using that score matrix or a matrix to find that minimum edits or find the minimum sequences. And so here are my references. And thank you so much for watching. Um, yeah, thank you so much for watching.